Okay, guys, we are back for the last part of chapter 4. And I mentioned earlier that I had a little bit of a scratchy voice, which has seemed to really progress throughout this day. So I'm going to do chapter 4 and then take a break and get chapter 5 up for you guys after I get maybe some warm tea in me. So probably will not be posting chapter 5 tonight, but I wanted to make sure that 4 was complete for you guys. Um, I don't really do a lot with this last part of the chapter simply because, for the most part, we are going to focus on the fact that we deal in microbiology mostly with prokaryotic species. Now, I told you we would cover some eukaryotic species because we're going to do fungi and we're going to do helminths. Uh, but as far as the cellular level go, I just wanted to remind you of what a eukaryotic cell is. Remember, it does have all of those organelles, the mitochondria, which is nicknamed the powerhouse of the cell, and the Golgi apparatus, and it's got the smooth endoplasmic reticulum, the rough endoplasmic reticulum. Remember, it does have a nucleus that is a specified, it has a nuclear membrane around it and an envelope around that with pores that allow regulation of inside and outside of that control center. When we look at eukaryotic cells, if you think back to anatomy and physiology, you may remember talking about cilia. Cilia are little hair-like structures that beat. I always think of a centipede. If you take a centipede and you turn it upside down, you have those 100 little legs going 90 miles an hour. That's what cilia sort of look like on the cell. They beat in unison together. And then some cells do have a flagella. Eukaryotic species don't move the same way as prokaryotic species. Let's think back to how prokaryotes move with a flagella. Remember talking about the run and the tumble, right, where they move in one direction and they flip or do a somersault or a tumble, and then they move in another direction and they tumble again. And we used to think that that was random, but now we know that there are receptors that we can actually manipulate or control that movement to a certain degree. Well, eukaryotic species can sometimes have flagella as well, but they move more in a wave-like manner, sort of like if you get down on the floor and do the worm. Someone did ask me in my class if I would do that, but that's a sight you don't want to see. So sort of like a wave-like motion for eukaryotic species to move. When we look at a cell wall, you guys know as eukaryotic species, you do not have a cell wall. However, plants have a cell wall, fungi have a cell wall, and so structurally, the cell wall of a eukaryotic species is actually very simple when it comes to comparison of those prokaryotes. We also see by comparison the plasma membrane very, very similar to what we talked about in prokaryotic species, except we differ in the types of proteins. Now remember, look down this list right here, and next to the bottom you're going to see sterols. And I told you there is one species that does have sterols in its plasma membrane, and it was which one? Anybody? Anybody? Oh, well, okay, let me tell you. It was Mycoplasma pneumonia. Remember, Mycoplasma pneumonia is that atypical cell wall. We still have movement across the plasma membrane in a eukaryotic species, except now we do have endocytosis. And remember from A&P, endocytosis is where you are able to take something from the external environment and bring that to the inside of the cell. Eukaryotic species, of course, do have the cytoplasm, which is that fluid-filled portion. Here is a list of all of those organelles that we are not going to cover. One interesting, just because of one concept that I do want to cover for you guys in this final chapter that has been a debate amongst microbiologists for several years, is the idea of protein synthesis and where it's found. We know that prokaryotic species have ribosomes as well as eukaryotic species. And I told you that with the prokaryotic species, They have that small and large subunit, that 50 and 30 subunit, that give us a total ribosome when we put those together of a 70S. Well, eukaryotes also have a small and a large subunit. The large subunit is a 60S, and the smaller subunit is a 40S. And when we put those together, remember you don't add them, but when we put those together, the full, complete ribosome for protein synthesis is known as an 80S. And the reason why I bring this up, as I said, is because conceptually, I want to talk about a theory that microbiologists have debated. And one of the theories is based upon this slide right here. When you look at a eukaryotic species, everywhere in that cell, you are going to find those ribosomes that are that characteristic eukaryotic 80S ribosome, except for in two places. One is in the chloroplast, and the other is in the mitochondria. Now, if you think back to the mitochondria, it's an interesting part of the cell itself. Mitochondrial DNA, for example, remember from AMP, is only maternal in nature. You are, by genetics, 50% your mother and 50% your father. However, mitochondrial DNA is 100% from your mother. And then when you look at the fact that the mitochondria is the powerhouse of the cell, it's where we get our energy. It does the most work, if you will, to give us that ATP by cellular respiration. But when I look at the ribosomes associated with the mitochondria of a eukaryotic species, and I spin those down, they don't break apart into that 60 and that 40S subunits. Instead, they break apart into a small 30 and a larger 50. Now you tell me what that sounds like. Does that sound like a eukaryotic species? No, it doesn't, right? What's it sound like? It sounds like a prokaryotic species. And so we started looking at the idea, or the theory, if you will, of what is called the endosymbiotic theory. Microbiologists believe that originally there was some form of universal ancestor, and that eukaryotic species, in order to function at a higher level, actually engulfed some of the larger bacteria that lost their. That some of the larger bacteria engulfed some of the smaller bacteria that then lost their cell walls and remained within those other cells to function and actually live one within each other. One of the things that you don't know about prokaryotes yet, because I haven't told you yet, is that they are excellent when it comes to making energy. Energy is all about how we are able to survive. And as far as prokaryotic species go, they're excellent. They actually make per cycle of cellular respiration 38 molecules of ATP. So they're very efficient when it comes to making energy. And so scientists believe, based upon this theory, that whenever these larger bacterial cells sort of engulfed these smaller ones, and they enslaved these smaller ones, and they started using these smaller ones to their benefit, and the benefit that microbes could give us would be ATP production. So why do they think that the mitochondria, therefore, is the one organ that the endosymbiotic theory represents? Well, number one, you just told me what the mitochondria does. It's the powerhouse of the cell. It's the energy producer of the cell. And that's what microbes do so well. They make energy. And number two, I told you that everywhere in the eukaryotic cell, we have protein synthesis ribosomes that are going to be 80S in nature, spun down, just centrifuged down into breaking apart into that large 60 and that small 40. But that's not the case for the mitochondria. In the mitochondria, it mimics that ribosomal content of what we see in a prokaryotic species. So based upon that, it is believed that at some point these bacteria were uptaken and, instead of destroyed, utilized and put to use for their benefit of the other cells. So I just wanted to mention that concept. I know I don't do a lot on eukaryotic species because honestly we don't cover that many eukaryotes here. And I know that you were exposed to AMP1 and AMP2 because that is actually a prerequisite for this course. So I'm aware that you guys were exposed to that in the beginning. So I know you have covered what all of those cells do, so I won't be asking you to redo those eukaryotic properties for the test. We're going to focus this chapter mostly on what we see with the prokaryotic species. So next up is the final chapter that will be associated with chapter 1 to, exam 1. Exam 1 is going to cover 1, 3, 4, and part of 5. So thank you guys very much. I will get back with you for chapter 5, and I hope you have a blessed night. See you later.